Good morning, everybody. So very nice that you can join me this morning for this presentation, where I'll be focusing on the use of metabolomics, particularly in biotechnology. Now, with these sorts of presentations, obviously there can be a wide range of audiences. Some of you may be quite advanced in the techniques, others of you may just be starting out. So what I'll try and do is to introduce things at a very basic level. So apologies for anyone who is learning stuff they already know. And then I'll go on to talk about some concepts in rather more detail. Obviously, metabolomics itself is a very big field, and I might not be able to cover everything that you're particularly interested in. So please feel free to ask any questions at the end regarding that, or indeed do feel free to contact me later with any questions. So with that, let me just go on to talk a little bit about what we're going to talk about this morning. So I'm going to spend quite a bit of time introducing some of the basic concepts of metabolomics. It's a field that exists within the context of other fields, and as we go through the presentation, I'll be making some references to that. I'll be talking about the range of technologies, analytical technologies that we can actually use to study metabolomics. And again, these sort of analytical technologies are very broad, and I won't be able to do justice to all of them, or perhaps even the ones I'm going to mention. So again, there's a lot of technical detail behind that. That you may want to go away and look up yourself, and there's lots of good papers around to do such things, some of which I'll be referencing. I'll then be talking about how we handle the data that we get from these sorts of very large experiments. That's a very critical part of metabolomics, and it's something which actually probably requires, in fact definitely requires, almost a lecture in itself. So again, I'll be only very briefly touching on that to give you some sort of flavour of what that is. Then what I'd like to do is focus the talk a little bit on the importance within biotechnology, and sort of particularly the areas that I'm working in at the moment. And then we'll also touch on how we can sort of extend metabolomics, and indeed we may not be calling it perhaps metabolomics at that point, but certainly the principles are fundamentally the same. Where we look at applications in bioproduction processes, again, these are very broad. I'll be making some general points and some examples, using some specific examples to try and illustrate that. And then I'll be most happy to take questions from you for considerable time, or as short as it happens to be. So, you might find it a bit strange that you're at a talk about metabolomics and the first slide you see is about the second law of thermodynamics. But basically, when we're talking about understanding or wanting to analyse the biochemistry of an organism, we need to understand what are the basic principles that are going on here that are essentially driving life and its biochemical processes. So some of you may be familiar with the terms of low entropy and high entropy. What this is basically trying to illustrate is that within organised systems, chemical systems, and of course biology is an organised chemical system, we have a state of what's termed low entropy. I can't speak this morning. Entropy. So this is where we're deliberately maintaining an equilibrium, well not an equilibrium, a state which is outside of the normal, if you like, ground state of the surrounding environment. And of course that's exactly what cells and organisms do, is they essentially gather up energy and they use it for their own internal processes. And when cells die, or organisms die, we can see the result of that natural decay of energy as a system moves to a state of high entropy, which is actually, if you like, the stable ground state. This isn't the same as, you know, active decomposition processes. But basically an organism needs to be able to regulate itself, to be in a state of low entropy in order to control its processes and control the function that it wants to do. So there are universal metabolic regulatory principles that all life adheres to. They're actually very simple, whether you are a bacteria, or whether you're a plant, or whether you're a higher organism. And essentially what you're aiming to do is to take in energy, often as much energy as you can, in whatever form that might be, light or chemical energy, if you're an animal. You then need to have processes which convert that energy into what you want. That may be physical structures, it may be activity. And then you need to use that energy to reproduce cells and reproduce copies of yourself. And the whole of biochemistry in every organism is essentially targeted towards that goal. And these processes that we need to be balanced, and they need to be controlled. And how organisms do that is basically the study of biology, but specifically when we're talking about the chemical nature of things, this brings in the field of biochemistry and eventually how we would use metabolomics to look at these processes in much more fine detail. And metabolomics and its associated techniques are attempts to dissect and to understand these basic tenets of what life is actually wishing to do. So, if like me, many of you will remember in your biology classes at school, large posters of very impressive looking biochemical pathways plastered up on the wall of your teaching lab. In fact, it was one of the things that actually stimulated me to become interested in biology and biochemistry, was this incredibly rich looking and detailed description that we seem to have of the biochemistry of life. And indeed, these sort of pathway maps were starting to be generated well before the discovery of the double helix. So biochemistry had an awful lot of understanding, apparently, of the details of the chemistry of life before we had anything associated with molecular biology. These pathway maps were worked out in the late 19th and first half of the 20th century, and essentially were test tubes experiments where enzymes and substrates were extracted from organisms and tested on the bench. But it's probably important, very important, I would say, that we don't get overwhelmed by this apparent grasp that we have of biochemical processes. As I'll go on to talk about later, you may look at these sort of maps and think that it's extremely easy to chart a course for engineering or changing these pathways, because they appear to be so well connected, so well understood. But the reality is that we don't know anywhere near as much about these sorts of interactions as it might appear from charts like this. In fact, one could say that very much the more we learn in modern biology and modern molecular biology, the less we actually know about these sorts of biochemical organisations. And indeed, many individuals and many organisations have embarked on biochemical engineering processes on the basis of assuming that these types of interactions that we can see here are things that are easily understood, easily manipulatable, and can be targeted to produce outcomes that we want. And the success of such what I would call naive processes has generally been not at all what the originators thought about. And I will go on to discuss why some of these reasons may actually be. So take a little step back here. If you like, the understanding or the interest in the chemistry of life, particularly related to humans, has a very long history going back to classical times and the ancients. For those of you who are interested in the history of biology, there are many references in classical literature of all cultures of smart people from the day recognizing certain things that we now understand to be fundamental and actually part of biochemistry understanding. Take one example, the Muslim scholar of the 13th century, Abu al-Nafis. I struggle to perhaps pronounce his name right. He was a very famous physician back in his time, and he made this statement, which I think rings very, very true today. That was, the body and its parts are in a continuous state of dissolution and nourishment, so they are inevitably undergoing permanent change. And I think those of us who understand about the sort of dynamic fluxes, the dynamic equilibriums of cells and biochemical systems, understand that there is a fundamental truth to that statement, even though it was made nearly 700 years ago. But skipping forward, the interest in using biochemical measurements to understand a bit more about processes starts within our lifetimes to quite some extent. In the late 1940s, Roger Williams, for example, was doing paper-based chromatography on saliva studies and was generating different profiles that he could see on these little bits of paper from patients with different types of diseases and psychological issues. If we fast forward then to around the 1970s, Hornin first coined the phrase of metabolomic profiling, and he showed using GCMS that compounds present in urine extracts could be measured and could be quantified. But metabolomics as a separate sort of nascent entity really started to kick off in the mid-1980s with works such as a group of Professor Jeremy Nicholson, then at Imperial, where they demonstrated that use of nuclear magnetic resonance spectroscopy was able to be diagnostic for body fluids for people suffering from diabetes. The chemical shifts of profiles could be seen to be statistically different. And it was then around the early 2000s and then ongoing till today that a real push has developed to try and catalogue metabolomes and indeed the human metabolome in the same way that there's been the effort to sequence the human genomes and other organisms. And sort of currently, although it's a very kind of hand-wavy figure, we estimate from post-genomic data that there may be something around 5,000 metabolites present in the human metabolome of all major cells and body fluids. But interestingly, the plethora of biochemical reactions is much more than that. Nearly 18,000 biochemical reactions may be possible, not necessarily in any one cell, but possible without the body. And I think this also hints at it's not just measuring the metabolites that are important, it's actually understanding how they interact that is really fundamental to what we are able to deliver. Sorry, slide. Yeah, that's fine. Okay. So, a real seminal paper in terms of understanding how we use and why we want to use metabolomics came with the sequencing of the yeast genome. This was the largest genome then to be sequenced, and there was an important paper that was sequenced in 1996, and some 4,000 genes found to be encoded. But a paper a year later really used that information and talked about the functional analysis of the yeast genome, and it was in this paper the term metabolome was widely introduced. And the authors make an important comment. The challenge is now to discover what each of the gene products does and how they interact in the living yeast cell. So what we actually see in this statement is that metabolomics is very much a post-genomic tool. We can interpret it based on and use it to interpret what actual genome sequences have. So in that respect, it doesn't exist separately. And these same authors were also able to demonstrate what we now recognize some of the main principles that we see in the metabolomic experiments. For example, they did some screening of yeast lines using rapid mid-infrared Fourier transformation spectroscopy. And they subjected this data to multivariate analysis to look for different groupings of yeasts that responded to different growth mediums different ways. So in this seminal paper, there was really everything that a metabolomic experiment actually requires. So if we think about how we define the word metabolome, it's really quite simple. It's essentially the derivation of metabolism and genomes. And we would define it as a sum of the reaction pathway intermediates that are present at any one time in a biological sample. And the study of it has very much co-evolved with our instrumentation, particularly LC and GC mass spectrometry. And as the power of these instruments and the power of data analysis has increased, so has our ability to do this type of work. Metabolomics exists within the basic hierarchy of biological control. So we have the genome, and then, then we have that is then transcribed into transcripts. The transcripts are then used to make amino acids, and there can be hundreds or thousands of amino acids in a particular protein. And all enzymes are proteins. So this cascade, if you like, controls the biochemical transformations that can occur in any given system at any time. And measuring these interconversions that these enzymes control is essentially what metabolomics is. Now, one question I'm often asked is what omics system should I use to study? There's not actually a wrong system to study. It depends on many things, your expertise, the availability of the equipment, maybe what's already been done and known in your field. But we can characterize some of these technologies having different properties we might want to use. So genome sequencing is now common. It's not very quantitative, and it's still on a large scale more expensive than some other techniques to do, vastly cheaper than it used to be. But it's not a quantitative technique. It gives you an idea of what could be there. Proteomics, which is the, if you like, the twin of metabolomics, studies a complete complement of proteins. One of the advantages of this is that protein sequencing now with post-genomic information is very good, so you can get functional information quite easily. However, it struggles to be quantitative, and it also, in any one experiment, struggles to be sort of as holistic as metabolomics, because the proteome is complex and often needs to be fractionated before you do the mass spectrometry analysis. Metabolomics is certainly faster to initially do than the other two omics, and it's more quantitative. But we have a big challenge with identifying compounds. In a typical first-pass experiment, you'd be lucky if you could identify 10% of the 4,000 or 5,000 peaks you might see. And that's a challenge which I'll come on to talk about later. They also, the omics also answer questions at different levels. So genomics is basically all you can do in a lifetime. Transcriptomics, transcripts change in minutes. Proteomics, protein levels change usually within minutes to hours. Metabolomics can change in seconds, which is also a challenge as well as an advantage in your study system. And overall, metabolomics can now be applied to a vast array of different investigations, as you can see displayed here, and I will be just touching on one of them as an example of this. So let's have a little think about some of the analytical techniques that are currently being used, some of the more important ones. Metabolomics is basically based around two, possibly with an extra third one, technology. Nuclear magnetic resonance is almost where it started with the work of Jeremy Nicholson and urine. One of the things about that is that it's very easy to put in samples relatively untreated. However, the sensitivity of NMR is an order of magnitude less than mass spectrometry, and therefore the range of metabolites it can measure in a typical sample is really quite small. But it's excellent for doing certain things like primary metabolites in urine and blood samples. Mass spectrometry comes in many flavours, which I'll talk about. And there's also an opportunity to use other sort of fingerprinting techniques, optical techniques like FTIR. The big thing that metabolomics has, unlike sort of using these types of technologies for other biochemistry, is that we need to interpret the data via statistical modelling because the data sets are often very large. And this practice you may have heard of is termed chemometrics. So mass spectrometers started off as very large instruments indeed, and as I mentioned, one of the challenges of them was the computational power that we had. Although you see the operator here in what we would consider now totally inappropriate PPE for a laboratory setting, the most important thing to note is the large computational framework you have to get any data. And you'll see a lack of any sort of VDU, it's simply a printout. And what I put this up to is illustrate there's not so much an issue these days, or even in the past, of getting the data, it's actually handling it that's been one of the biggest challenges for the community to solve. One other question I'm often asked is what mass spectrometer should I actually buy to do this work? There is no one answer for that. Each instrument, and indeed each vendor's instrument, has its particular strengths and weaknesses, some of which I've highlighted here, such as accurate mass instruments are very good at discovery type of work where you don't really know what you're looking for and you want to calculate under empirical formulas and do fragmentation patterns. If you have targets, then triple quadrupoles, which are targeted instruments, can be tuned to specific metabolite panels of interest. Gas chromatography will do all these things as well, both targeted and discovery. And it obviously has a slightly different overlap compared to liquid chromatography in terms of the samples it can actually handle. A quick flyby of my technique of choice, the mass spectrometer. Here, this is an example of a time-of-flight instrument, and if we cut it away, the only points for this talk we really need to understand is that there's some way to get samples in, which requires ionization of the samples. The molecules enter the instrument, and they can be filtered and fragmented in different ways. Depending on how you want to study the molecule, and at the end there's a way of detecting its abundance and its mass, and in this case it's a time-of-flight detector. Understanding mass spectrometry will be a whole nother talk, so that's all you're going to get today for that. What's more important, probably for the context of this presentation, is to understand how we can capture the metabolome at a fundamental level. So it's not a matter of simply grinding up a sample and washing it with a bit of methanol and chucking it into your instrument. The metabolome is incredibly rich, and all analytical techniques struggle to deconvolute that sort of rich information. So generally we would try and fractionate our one sample into several different fractions and run them in different ways on different platforms, which would target different types of metabolite pools. You could easily end up with 5 different data sets there, therefore run 5 times on the same or different instruments, which would be very large, and often we try to reduce this down to 2 or 3 acquisition settings, which will capture what we really want to capture. But it is potentially an important part of method development. Okay, now the sort of data we get out of mass spectrometers is incredibly detailed. We've got an example here of one sample chromatography run with time on the bottom here and abundance on the x-axis here. If we home in on one peak that we think perhaps if we were just doing chromatography might be one compound, we see it's not. There are many mass spectral peaks within that, and within those regions of the mass spectrum we can zoom in again and we see a very complex mass spectrum that we need to quantify and that we can use to identify individual compounds. It's very data and computationally intensive process. Thinking about trying to identify compounds, there are several different strategies you might adopt. There are electronic libraries available out there which have spectral information that you can look to try and identify your sample from. These include things like the Metlin library, and for GCMS, the standard NIST library. However, it still remains that probably if you want to get the most power out of experiments, you do need to go down the process of building an individual library for your particular, certainly instrument, your particular laboratory. There are, as I say now, an increasing array of electronic versions of these, but basically you're talking, if you want to get something like 900 or 1,000 plus human metabolites in your system, you'd be talking to buy them from somebody like Sigma, probably around 20k to get your library, and then obviously a lot more investment to actually run and build that library. So it's not maybe as accessible as simple genomic sequencing is, for example, or identifying proteins, which we can go to electronic databases for. There's a cost in terms of how far and how deep you want to go down into the identification of molecules. There are different levels, going from we just want to see that we think it's a feature, all the way down to actually identifying what that actually is. And we have different workflows to get there. The take-home message is basically, as you try to be more confident in what you've got, there is increasing time and increasing cost to do that. So you need to tailor how far and what you identify for how much time, how much money, and what your specific need is. So I mentioned that data handling is important, and I will now introduce a few little concepts for that for you. If we think about the data that comes out of a mass spectrometry, high resolution, untargeted global metabolomic experiment, it might look something like this, with each of these traces being an individual sample that you run, and you may have dozens or hundreds of samples. We have to take these individual traces and we have to actually align them so that we get the same masses and retention times aligned in columns, so that we can then compare one sample to another. A lot of that will be noise, and we have software and algorithmic routines that get rid of a lot of the stuff that is just there as background and not our interesting things. Eventually, when we go through that process, we end up with peak tables which have masses of our particular extracted peaks and compounds, and then we have to do something which is a little bit outside this talk, which is then to use natural abundance of isotopes to help us try and identify what peaks may come together. But there's basically a workflow that you go through merely to get your data out into a tractable form before you do any type of compound ID or anything like that. Again, how you do all this would be the subject of a whole talk. However, I think all I will say is that unlike when I started 15 years ago, there's now a plethora of good software from both the instrument vendors and from freeware communities of very good quality, which is allowing you to process your data. They come in lots of different flavors. They can be matrix-based. They can be like this example here, which is a node-based workflow. Okay, so your data is processed through different nodes, giving you eventually an output that you're interested in. And all this, many of these softwares are now in Windows GUI-based environments, so you don't need to be a coder in R or Perl or MATLAB in order to use them, which I'm not, and I don't have time to do. I just want the data out, and that is no longer the handicap that it used to be when I started. We can then think about how we might want to take that data on a little bit further. So here's an example here of round about 20 odd samples that have been analysed, and what we've now got here is your samples along here and the relative abundance of the extracted peaks shown in this heat map here, and in this case we found about 420 compounds. Now if we want to understand what the patterns in that data, we can't try and plot 420 different compounds against each other to see how they change or what groups have different effects. So we then actually move on to the realm of multivariate statistics to do that. Again, that would be another presentation to speak about how we do that. But essentially what you're trying to do in something like, let's say, a principal component analysis, is to consider that we've got multiple dimensions, hyperdimensions of data here. Okay, we can't see or understand that because we only live in a three-dimensional world, but mathematics is capable of doing calculations within multiple infinite dimensions. And something like principal component analysis essentially does what you might do with just a hand regression of a line if you're plotting x against y. It tries to plot what are called eigenvectors through your data in different ways that can then be more tractably visually displayed, such as in these examples here. We can take our individual metabolites and we can convert them to a heat map, so we can see what's going up in red and what's going down in blue in different sample types within different treatments. And we can do simple things like clustering analysis, which groups by similarity of the metabolic profile. We can also do things like principal component analysis here, where we have XY plots, where we plot our individual samples here, and we may look for outliers that in this case would have a different metabolic profile to the to what's going on with the main group here, in this case some E. coli. And there are statistical tables behind these things which will then give us clues as to what are the most important metabolites that are driving this type of separation. And it's this workflow that delivers biomarkers of interest, for example, for process control or for clinical studies or for anything. So we move from needing to understand how everything changes to how just a few metabolites are the most important ones affecting the process of interest, and we do that via the multivariate or other types of statistical modelling to essentially filter that data out. Once you've got metabolomics established in your laboratory, you've then got different flavours you can think about working at. You may want to do rapid screening of large samples, maybe animal populations, human populations, plant populations, cell populations. We can do that rapidly by what are called fingerprinting techniques, okay, where we're trying to move quickly from our data acquisition stage to plotting some of these types of statistical relationships, with really not much interest in actually identifying what the compounds are. We just want mathematically significant patterns. If you want to be mechanistic, so to understand what a particular metabolic pathway is doing, then we would probably want to do much more optimization of our data acquisition techniques, whether it's mass spectrometry or NMR. We would then want to use our data modeling to filter out compounds that we then want to try and identify to understand what they may be. Another important thing is that metabolomics does not and should not exist as an independent entity in itself. So, for example, if you've already got some gene expression data, you can synthesize that with metabolomic data by trying to map the two things to known pathway structures, overlay the data, and look for functional correlations in that sort of way. So, I'm now going to change tack a little bit from generalization to a little bit more specifics, and I'd like to focus our thoughts on the importance of metabolomics in modern biotechnology. There's three important things we need to remember here. That we think we know quite a bit, but we probably actually know rather less than we think we know, and there's also a lot that we really don't know, and we sort of need to understand what we don't know in order to set the limits of what we can actually do for biotechnology. So I already touched on this idea of the impressiveness of metabolic maps. This here isn't a metabolic map, it's actually a map of the London Underground. But I put it up to try and demonstrate a salient point. That is that maps look impressive, but they're just your possible journeys you could take on an underground. It won't, for example, it won't tell you when you have to arrive to get your train, and it certainly won't tell you how the trains are controlled. And in this area where we have an awful lot of seemingly very powerful post-genomic data, and we have synthetic biology, which is very easy now to make manipulations to biochemical pathways, it can be a very attractive tool to use those technologies and use metabolomics in order to try and design the metabolism in ways that we want. However, we do have to be extremely careful about this, as I've already illustrated, that actually fundamentally what we know about the metabolism is nowhere near as detailed as the forensic dissections with genetics that we can now do on that. The truth is that actually many approaches to do direct or smart biochemical engineering have mostly led to unexpected or unintended results. And this is not surprising. When you look at maps like this, you can see that essentially everything is related to everything else. Okay, so if you push the system in one place, don't be surprised if it tries to push back in another, especially because organisms like to try and maintain equilibrium and don't necessarily want to do what you want them to actually do for you. So often what we see are these what are called emergent properties. So these are properties of how the system responds when we look at it in a very wide global context. And of course, metabolomics is great for that because we can cover, not relatively easily, but we can cover quite tractably large portions of the total biochemistry of an organism. So if something small is happening, we've got a good chance of seeing it in our metabolomic profiles and by our data and statistical understandings. So metabolomics can contextualize what actually happens when we try and do engineering-based biochemistry. In terms of bioproduction, we don't have to use metabolomics just to do genetic engineering. We can also use it very successfully in bioproduction-only contexts. So, for example, here, when you're scaling up production of microorganisms from almost the bench scale to industrial scales, we might actually want to know what changes and what's going on there, and metabolomics can be great for that. Now, this slide is kind of my mantra, and it's a little bit wordy, and I apologize for that, but I will sort of read out some important parts for you. The way I see it is microbial systems, microbial cells are basically open, what we call open systems. They are looking to produce more of themselves. They take in biochemical products, and they take in heat inputs. And we actually also input other things such as carbon sources and micronutrients to them. But often, particularly in traditional kind of process control of bioproduction, we just sort of measure the nutrients, or we measure the final outputs, such as the growth rates, the pH, the sugars taken up. And all we try to do is to kind of use these very limited measurements in order to keep a process within specified limits. However, what we really should be thinking is that there's an opportunity to use the cell as a biosensor, because if we can measure the whole of the systems, biochemistry systems of the cell, we actually can look at the critical feedbacks that are going on within that system. And we can use hundreds or thousands of metabolites in statistical models to then try and optimize how we control our production system. And this actually holds whether your production system is a microbe or whether it's a herd of cows. It really doesn't matter. It's simply scale and accessibility. And if we can access hundreds or thousands of metabolites, we can then build quite detailed models of how processes work and how changes to processes, not genetic changes, but actual production processes, scale up, scale down, changes in nutrients, will actually affect our system, and we can predict forward as to what we might need to do to bring our system back to some sort of economically operable state. So, these are sort of plots we might get for typical outputs from bioreactors, things like pH or nitrate levels. They go up and down over time, in this case days here, and they may follow a profile that we can sort of recognize by eye if we are expert with the system. But these properties will change as we scale up, as things like, for example, pressure changes, mass transfer of chemicals and gases change, and forces like sort of shear forces as you have stirrers within these systems will actually change. And there's not a simple linear scale up from a small bioreactor to a big one. Metabolomics and its hyperdimensional data is actually one of the ways that we can try and build models to forward predict or even backward predict if we want to go to sort of fast screening techniques that are cheaper rather than running our 100,000 litre bioreactor and going, we tweaked something, it didn't work, and it cost us 50 grand. Do it on a lamp scale and use metabolomics to link these different levels via modelling is a smart way to proceed. We follow a sort of very generalised and typical process when we go through metabolomic experiments, whether it's production process engineering or whether it's some other thing. And that essentially is we get a sample, we prepare it, we capture it with an analytical technique, we take the data and we do some modelling. We then might want to use that data as a way of sifting through important pathways and plotting those and understanding what they might be. And we can then use those outcomes to change a process or redesign our organism. And you see this is the cycle, it never ends. You can keep going round and round it to iteratively improve what you do, and one thing will actually inform the next stage of design, such as your results might then dictate what type of sample prep you then might want to use to target particular types of metabolites that you now understand are interesting for your process. And as I mentioned, statistical modelling is very important for this. In terms of production processes, typical types of analysis we might do would be sort of differential analysis, where we're looking for groupings of our data based on different treatments. We would look at statistical plots to understand which of our measured metabolites are the most important, looking for high and low responses in them that drive in this model. And we can even do simple correlation regression analysis. For example, if we wanted to increase the stirring rate of a microbioreactor, we might see that has a positive impact as the stirring rate increases. We can build a model that actually shows a linear correlation to that. All these things are entirely possible. Of course, the Uber experiment would be to take our metabolomics workflow and embed it fundamentally within our basic economic operation of a process. So our model has outputs, and we can take that to understand how it might affect our bioproduction, may affect things like downstream processing and product control. We can take ideas of genetic networks and engineering to both inform our process design that we would use metabolomic data to actually monitor, and also sticking it out to actually understand what are the most important metabolic networks. And actually as well, we can also take all this prediction, which essentially can be boiled down to how much and how cheaply can we do it, and we can get an idea of what may be the economic value for our customers. A common area within microbial bioproduction is to try and optimize energy production. This can be one of the biggest expenses to your system's input. And in single cells, we know a lot about the simple pathways that goes on, which are very tractable to metabolomics. And we can look at various parts in the process of something like energy production, where we may want to increase or perturb things to drive systems to either aerobic or anaerobic states. All these things are very well indeed served by measuring the metabolites that are occurring here, both in the cell and that's excreted into the medium, rather than simply measuring fairly blunt outputs like optimal density as cells increases, or single parameters that we might want to measure and then try and extrapolate it to what actually is a complex pathway. No, far better to actually measure directly what you want to understand is changing within your system. Now, in the last few minutes, I'm just going to jump a little bit further into kind of pushing out metabolomics to other areas of biotechnology production. That may be food feed, that may be pharma, it could be high value products, it doesn't really matter. But an important aspect is to understand that the landscape in which we try to now understand developing technologies in bioproduction systems are changing. We have things like, instead of batch reactors, we're now moving to continuous bioreactors. Modern types of microbial and single cell production systems can operate very quickly, so we need rapid analysis. We need to get feedback of our data so we can control the system with computer systems. And often we may want to understand how the downstream processing can be optimized by tweaking the bioreactor processes. Metabolomics, as I hope I've introduced to you, can operate at all these levels to deliver data for these needs. And even if we go on to more kind of advanced ideas of process analytical technology, such as PAT, where we are trying to understand how we can monitor systems as we scale them up, scale them down, and validate them for different regulatory purposes. Again, and I won't go into too much detail, regulatory authorities like the Federal Drug Administration is now completely embracing the idea of using chemometric models and metabolomic data so that you can build acceptable types of arguments for regulatory use. Okay, these are all things now where we're starting to move away from, if you like, single gold standard measurements to say my product, my drug is safe, it's been produced in my bioreactor in a safe way. No, we're actually going for a much more holistic approach, which is a systems-wide characterization, and we're saying, using metabolomic data, if we have a broad profile of our metabolic production system that we compare between good batches and bad batches, we can use that in order to release our product and to show that it's safe and that its efficacy is actually still intact. And introduce another term from bioproduction system, this idea of multi-attribute method analytics, so-called MAM. Again, a whole talk could be made on this. Basically what it boils down to is, can you measure the same things throughout your production process, throughout your engineering, all the way out to product release? Metabolomics can measure hundreds or thousands of things, so you can get profiles of how your product is produced and how it changes during storage and formulation and all these other things, so that you can meet ideas of quality control and quality by design. So, if what I've talked about is interesting, you'd like to learn more, then myself and ANSYS can help you in many different ways, from basic mass spectrometry training consultancy all the way to understanding what these multivariate analysis techniques are, both for beginners and for more advanced users. And we can also help you to apply this learning, this understanding, for a whole range of things, whether it's designing and optimizing your system, or and the clients I work for at the moment are using this technique for submitting studies for FDA regulations, for quality control, etc., etc. Metabolomics is a great technique for using that, and we can help you to meet those aspirations if you really want to. If you want to learn more about chemometrics, we've got an upcoming webinar course on the 7th and 8th, which I'll be delivering, so please log on and register for that if you wish to. And yes, with that, I will bring my presentation, my whirlwind presentation, to an end, and I'm happy to take any questions from the audience or the moderators. So thank you very much for your time, and I hope it was useful for you.
Wonderful. Many thanks, Simon. I see that we've got one question from Mohammed. So apart from the NIST library, can AMDIS software also be used?
It can be. AMDIS was designed primarily for GCMS software, and its algorithm for spectral and peak deconvolution is sort of optimized to that, but it can be used for deconvolution LCMS data as well. So yes, you can use it, and you can use it successfully. It's perhaps much less flexible and more difficult to get immediate outputs from it in the same way, sort of interactive outputs, as modern software. But it's a good tool, and many people, including myself, still use it.
Thanks, Simon. Are there any other questions, anyone? So you can put them into the Q&A box, or also into the chat box as well if there's got any other questions. So Simon, talking about deconvolution software then, is there any other types of deconvolution software that you can recommend?
So there's a whole plethora of sort of deconvolution software that individual vendors provide for their own instruments. Most of them are pretty good, and you can learn and use those if you've got that particular instrument, that data output. There's also a really nice selection now of community software that is able to do that. One program that I'd recommend people go into is something called MS Dial. If you put that into Google, it's literally MS and then DIAL. You'll come up with a whole organizational web page which is developing a sort of universal data analysis and key metrics package for the community that accepts all major vendors' data into that. And it also has some basic statistical testing within that, like principal component analysis. It's a very nice piece of software, has massive support and is being developed almost daily. So MS Dial, check that out and start learning it. Again, it's something I use quite heavily.
Thanks, Simon. How about other open source software that is quite useful to use for the software packages? What do you use currently?
Open source wise, again, MS Dial I use quite a lot. If you're talking about chemometrics type of approaches, there are open source things like that which are available if you hunt around the internet, free add-ins for Excel and other things like that. Probably R is the biggest freeware resource, and there are pages such as the Bioconductor organization. Basically, you can download R. It's not particularly a GUI interface, it's much more typing in a command line, but it's very powerful and it's free and it can have nice data visualizations. I'm a bit lazy and I tend to decide it's easier to pay a bit of money and buy some specialist statistical software to get where I need to get. But if you're not able to invest in that, then I would say have a look at something like R, which is quite good. And there's a whole community that's been there for 10, 15 years, giving you a full workflow for taking raw mass spectrometry data, getting your peaks out of it, running statistical tests, all that sort of thing is available if you prepare to learn a little bit of R in order to run it. I also use something called KNIME, K-N-I-M-E. That was that node-based example where I showed you a workflow where you can sort of string together different operational tasks. That's a free platform, and there are some mass spectrometry and metabolite workflows already being freely published for that. So that's more GUI-based, less command line. You don't need to know what the nodes do. You can just plug them together and work like that. That's something I've learned in the last few years as well.
Great, thanks, Simon. Are there any other questions from anyone else? Any other audience members? Anything in particular anyone was interested in listening to this webinar that they are thinking about doing in the future? No? Okay, so I'd just like to bring this webinar to a close then, and thanks, Simon. We all give you a big virtual round of applause, I believe. So thanks a lot. And if anyone has any... Oh, there's one question popped up from Camellia. What do you think about the FIEN library?
Yes, for GCMS, I use that extensively. I think a version of the library sort of might be free, but generally it's sort of packaged with a vendor's software. People like Agilent and other vendors have a plugin for that. Yeah, it's kind of the standard library I'd get for doing metabolomics for GCMS without a doubt. I would also say the MS Dial program I mentioned now has a very comprehensive EI GCMS library in there, which is probably as good, if not better, than the FIEN library. In fact, it actually is part of a lot of the FIEN libraries actually in there, because Oliver FIEN actually contributes to MS Dial heavily. He's one of the drivers behind it. So that's a great way of getting EI spectral data. Essentially, if you don't want to pay for it, get a library for free. But it won't work for LCMS. That's more specialised, and you tend to have to do individual searches for that on web-based platforms.
Great, thanks, Simon. Any final questions from anyone else? There's one, that one just popped in just now, so I just want to give everyone the opportunity to ask any final questions before we bring this to a close. No? Okay, well, thank you very much, Simon. Hope you all enjoyed the training course today, and as we mentioned, in the beginning of December, Simon will be teaching it's usually 1 day face-to-face course, but virtually we're teaching it over 2 days as 2 half days on chemometrics, and a lot of that will also be applicable, of course, to metabolomics. And then early next year we'll be teaching a 1 day absolute basics of a metabolomics. We'll then focus far more on that, and I think, Simon, it'll be kind of expanding on what you've been talking about today.
Indeed.
Absolutely. Wonderful. Thank you very much, everyone.
Thank you very much, everyone, and hope to see you again at some point. Thank you.